All right, so we started the recording and we're gonna start with presentation from Shri. And then we will start proteins, milk proteins. Go ahead, come on. Oh, wait a second, I need to give you, but you can come. Sorry, Shri. All right, I'll give you one to evaluate yourself after you're done. Okay. And write your name at the bottom to evaluate yourself. For Emily as well.
No, this like was a little question that I was like, I think it was super.
What was it?
It was like, I forgot that there was two placings instead of one.
Yeah.
Like, it's just, I forgot the second one.
Well, then I hand everybody behind.
Yeah.
24. Did you take one for the second? All right, we are ready for you. This is forward, backward, and then if you want to point, maybe a pointer, and that's for you. Okay. I did say 15 minutes because I think then it was little.
Yeah. Okay. Hello, everyone. This is my presentation on enzyme kinetics. So the topic of my paper was, and also the objective of the paper was the enhancement of fatty acid flavor release for low-fat cheese using carrier immobilized lipases on oil water Pickering emulsions. And before I dive into the paper, I want to break this objective down because Pickering emulsions, immobilized lipases, I just want to talk about what they are. And so, as we learned in class, in simple terms, an immobilized enzyme refers to an enzyme that is confined to a support. And this often enhances its stability, efficiency, it improves reusability because it holds the enzyme in place. So it's easier, for us to take it out once it's done using. And it improves alignment of the enzyme and substrate at the active sites. And yeah, overall, it just improves efficiency of the enzyme. And then Pickering emulsions. So these are similar to normal emulsions in that, they still have a dispersed phase, a continuous phase. But the difference here is that instead of using amphipathic molecules with nonpolar tails and polar heads, here we have solid nanoparticles which stabilize and reduce surface tension at the interface. And so going back to the objective, I guess the question is, why do you want to enhance free fatty acid release in low-fat cheese? And so I guess it's a well-documented fact that increasing, dietary fat intake, it leads to increased likelihood of obesity, among other things. And so one of the many solutions to this adopted by people today is that, well, I guess I ruined the animation, but I guess one of the solutions to this is that people are opting for low-fat food products, which obviously helps them stay, healthy. But then the big drawback of this is that they often don't taste as good, and so that's kind of why this paper is trying to figure out if we can, solve this issue as well for low-fat cheese specifically. Oh. Yeah, and so I guess previous attempts looked at by this study and other studies, they've looked at fat replacers like gums, they've looked at adding additional flavors, all of that, but it's had limited success. And so, just relying on free lipases to break down the triglycerides and release free fatty acids is not enough for these products because they have less triglycerides to start with. And so one approach was to maybe add additional enzymes or modified enzymes, which can kind of improve activity and just add to more free fatty acid release in this product. And so this study, I guess, focused not just on adding lipases, but adding them in Pickering emulsions. And this is because most hydrolysis, as we know, occurs at the surface. And so, as per literature, improved activity can be seen if the substrate is in the form of emulsions or micelles. And that's basically because the contact area increases between the enzyme and the substrate. So that's why they used emulsions. Moving to the experimental design. So the first thing I want to talk about is how they made the lipase and the nanotubes. And so they did this by using alpha-lactalbumin, dissolved in PBS, cross-linked with calcium chloride, and they used a protease as well. And here, I guess what this BLP, and they incubated this for 30 minutes. And so here during incubation, what's happening is the BLP is reacting with specific amino acids, and it's helping it assemble into those nanotubes. And then that's when they add the glutaraldehyde, which along with the help of calcium helps form like cross links along the side chains of the proteins. And so that helps complete the nanotube structure, if that makes sense. I don't want to go into too much detail because it's not like needed. But that's basically kind of how they make the nanotubes, is using proteases and breaking them down. And then they incubate that again, and then finally they just separate them from unimmobilized lipases and just make a solution out of it. Next, I'm going to talk about how they made the emulsions. And so to test basically whether Pickering emulsions do release more free fatty acids, they did this. And so they basically made a 2% weight lipase nanotube solution, where that would be the aqueous phase and the sunflower oil would be the oil phase. And they actually made two solutions of this. The first one, they used a high-speed hand mixer, which resulted in larger droplets. And the second one, they made small droplets using an ultrasonic cracker. And so this, I guess, in a way, they also wanted to see if the size of the droplets impacted how much free fatty acid is being released. And so along with these samples, they also had an emulsion with free lipases, which were not immobilized. And then finally, they had a non-emulsion based system as well. And I won't go into too much detail about the actual cheese making because that was pretty standard. But the thing I do want to talk about is that the previous methodology was to test if Pickering emulsions are, helpful in increasing activity. And once they did actually prove that, they made Pickering emulsions for the cheese. And so the only difference in this was that instead of sunflower oil, they used the triglycerides from the cheese, but the rest of it was the same. So the way that they did this was that they separated the milk fat by centrifuging, and then they used that to make the Pickering emulsion with the oil phase as the triglycerides. And then they just added that to 1 liter of defatted milk. And so and then they followed the regular cheese process making for that. And so there were 4 cheese samples made: full fat cheese, using like 4 different types of milk: full fat, non-fat, low fat with the immobilized enzymes, and then low fat cheese with just regular free lipases. Moving to the methods of analyses for the immobilization and for the Pickering emulsions. So for verifying if the nanotube process was actually successful, they measured these 3 things. So they measured the absorption of the tubes itself, which would prove if immobilization occurred. They measured the surface charge and lipase activity as well for the same. And then to characterize the emulsion itself, they did size distribution, they looked at the morphology, and they also looked at the structure of the droplets. And then finally, for measuring how much FFAs were actually released, they did a titration. And then apart from that, they also did other like just normal composition and texture analysis on the cheese itself. So yeah, it's pretty standard. I'm not going to read through this table. But moving on to the first set of results, which was for verification of immobilization. And so here, first, we're going to look at the morphology of the nanotube itself. So here you can kind of see that all of the tubes are covered with um, the lipase globules, with the help of the glutaraldehyde cross-linking agent. And so basically that shows that their process was successful in immobilizing the lipases. And then this second image shows the absorption spectrum. So the lipases were labeled with the CY5 markers. And so that marker has an it fluoresces the maximum intensity is usually seen at about 670 nanometers, I think. And so you will see that the red and the green peak, which is for the lipases immobilized on nanotubes versus just free lipases, they're pretty much the same intensity, which kind of shows us that they successfully immobilized all of the lipases and there was no like free lipases left in the solution after this process. This figure shows the zeta potential again for further verification. So the lesser negative value seen for the immobilized nanotubes was associated with the side chains of the protein gaining more positive charge once you immobilize them. And so I guess this kind of just again verifies that yes, immobilization was successful. Lipase kinetics. So this, a lot of this is what we learned in class. They measured the Km and the Vmax for this study, and then they used the Lineweaver-Burke equation to kind of differentiate between the activities. And so this is what they used to measure lipase activity on this next slide, which is this figure. Basically, it shows us that there was increased lipase activity compared to free lipases. Which if you look into more detail is because of this. So if you remember Km, it basically tells us about the affinity of the enzyme to bind to the substrate. And so if the Km is lower, that means you need a lower substrate concentration to reach the same Vmax. And so as you can see for the immobilized enzymes, the Km is much lower than the free lipases. At the same time, Vmax, it tells us how quick the reaction is. And so with a lower Km, the Vmax is also much higher. And so that ultimately tells us that the immobilized lipases did actually, I guess immobilization did actually improve efficiency and reactivity. And this is attributed to, again, the larger like contact area between the enzyme and the substrate, which is the triglycerides. And so yeah, these three things kind of verify that immobilization was successful and it is solving the purpose of enhancing enzyme activity. So the next set of results is looking at the Pickering emulsions. We want to see whether that, actually improves anything compared to regular emulsions. This is the only part of the study that I thought wasn't as well explained because here, so they kind of show us like CLSM pictures of what the Pickering emulsions look like. And over 90 minutes, you can kind of see a little coalescence, but they don't show us these for regular emulsions. And so I don't know if, a 90-minute period, like if this is a good time, like, is this more stable than normal emulsions or less stable? It's not a good like side-by-side comparison. So I guess this image for now just is good for us to see what the emulsion structure looks like in the final product. The final piece of results is looking at the actual free fatty acid release from the cheese. And so here you can, I guess the first two pictures just kind of show the size distribution again. So because they used, the hand mixer versus the ultrasonic mixer, you can kind of see the ultrasonic produce more homogeneous and more smaller particles, whereas the hand mixer had a mix of, both small and big, and it was less homogeneous as well. But this figure is more important because this figure actually shows us that both the immobilized or nanotube solutions, they definitely release more free fatty acids than the other two. And surprisingly, not surprisingly, but also they also saw that the smaller emulsion size also improved lipase activity, which could be because not only are you improving like the contact area between the enzyme and the substrate, but also the starting materials is the same, like the amount of fat you start with is the same. So you're basically improving efficiency of the reaction. And this is the final set of results, sorry. So this is when they actually measure texture of the cheese. And so here you can kind of see the green is casein, red is fat, and blue is the lipase nanotubes. And so the G figure with the pink is actually just superimposed red and blue. And so as you'd expect, most of them have fat except the non-fat one. And that kind of just shows us, it shows us the distribution. But up here, we can kind of see that um, that as the fat content decreases, the amount of free fatty acid also obviously decreases. But then if you look closely at just these two, if you compare them using immobilized enzymes versus free lipases, there obviously is a pretty significant jump of amount of free fatty acids released. And then in terms of texture, I just want to say that it's a good thing that they did not see much significant difference in full fat cheese versus low fat cheese in terms of all of these properties. And then if you just look at these two specifically, again, you don't see a big difference between if you add immobilized enzymes or you don't add immobilized enzymes. So that's a good thing because it's not really affecting your sensory properties of the cheese while it's improving activity. So in conclusion, the paper concluded that nanotube immobilization increased affinity of the lipase and reduced the potential energy barrier. It improves stability of Pickering emulsions and improve catalytic efficiency. The nanotube lipases could release more free fatty acids than free lipases, starting with identical milk fat contents. And yeah, when added to low fat cheese, it promoted free fatty acid flavor release as well. So a few critiques. I thought this was a really good paper because they had a lot of scientific evidence just for one objective. And they had a lot of controls at various steps as well to eliminate as many variables as they can. But the only drawbacks I saw was that the morphology, as I said, did not provide us with other emulsion morphology. So I don't know if a 90-minute coalescence time is good or bad. And glutaraldehyde is toxic. It's not food grade. So I guess that really brings the entire study scalability into question. And I know it's been used for biodiesel production for oil esterification, but I don't know how they would do this for cheese making. So with that, I guess any questions?
Yes.
Yeah.
So I know in the background says that it's like the whole question is to help flavor release in low fat cheese. You showed me structural data, but is there any any stronger evidence that it helps with the flavor release? I know in one of the methods you said there's TCMS, or do they equate the rate of free fatty acid into more flavor?
No, so I actually forgot to ask Pam about this. But that was going to be one of my critiques is should they have done a little more actual flavor analysis, because the way they used GC was only to find out or to quantify how much free fatty acid was released, compare that. But they did not yeah.
Maybe because.
Yeah. Oh, yeah. So the flavor in cheese, according to literature, comes from presence of free fatty acids. So if there's more free fatty acids being released from the triglycerides, there will be more cheesy flavor. And so that's what they're trying to do is have more FFAs released from a smaller amount of triglycerides.
Okay, so based on that assumption, which is literature, you can kind of.
Yes, yeah. And so that's what they were trying to measure or that's what they're trying to prove is that using immobilized enzymes are able to release more free fatty acids compared to just letting it be and using the free lipases already present.
Okay, and you're saying is that inherent to the full fat cheese, full fat milk?
Yeah, free lipases are naturally present.
Okay.
They didn't add anything to it.
So it's fat cheese versus low fat cheese. Do you think there's gonna be
Is that a
interference?
specific graph you're looking at, or?
No, I think this is a clarify. Did they quantify like this activity inherent to the, no? Because when you
Mhm.
immobilize like things from outside, what about the already specified thing?
So I think, no, they did not do that. They controlled the amount of fat to start with. That's why they defatted all of the milks and they added the same amount of fat in each of them. But
Like this.
yeah, but they did not control or they did not provide what the free lipolytic activity was.
Or did they add the like this?
No. No.
Okay.
Yeah, that's
All right, go
that's a good catch.
ahead. Yes, okay.
No, I think it was ripened for 2 weeks, I want to say. So they tested after 10 days, I think, to be specific. Yeah.
What kind of cheese is it? Fresh cheese and processed cheese or what kind of?
This is fresh cheese. Like,
Like
They-
the curd.
yeah, yeah. I don't know, is 2 weeks less for, okay. I'm not sure usually how long they age for. Okay.
So you said that they like didn't have any comparison images or like a control for the pickering emulsions. Like on what basis did they conclude that it was like effective?
What was effective?
The because I think you had said at some point that like the pickering emulsion was like better at.
No, so that's what, like, I don't know how they concluded the emulsion itself was more stable, that Pickering emulsion was more stable than regular emulsion, because I don't know how long a regular emulsion takes to coalesce in this matrix. So, because they had enough evidence for everything else, like to prove higher activity, to prove more FFA release, but I don't know how they concluded.
It was 90 seconds, or no?
90 minutes,
90
yeah.
minutes.
No, yeah, no. I guess it was just like like can you remind me like what was the point of the pickering was just to increase the surface.
Because that, according to literature, improves enzyme activity.
Finding
Yeah. Oh no, yeah, Pickering emulsion is because it's more stable. So apparently it doesn't coalesce as quick as if they used a regular emulsion, but I don't know how.
Yeah, like,
Quick.
I was gonna say like if that would
Yeah.
be my takeaway.
Yeah.
Yeah, but 90 minutes, they could last pretty bad.
Yeah.
Yeah.
Other questions? So basically, they use the toxic bacteria.
Yeah.
So what is the relevance of such research?
To show maybe that immobilization could help enhance enzyme activity, maybe. And like, because I guess the only thing that's making this not scalable is that cross-linking agent. So if in the future they are able to substitute that for another cross-linking agent, then maybe, yeah. Yeah. I'm trying to think back to Vichi's question. I feel like so I wonder if they corrected. So like you see the green line that is showing FFA release with just free lipases. So I know they don't explicitly say if they did it, but I wonder if they corrected the values for that, for like the other lines, the ones with the nanotubes. Because they did have like that as a control.
Is that what in the cheese matrix or like they prefer like a separate?
Yeah, so the difference between that's why I didn't know how to best show it. Like the cheese making methodology was kind of confusing. I had to go through it a lot of times. And so the green one is basically the exact same process as the red and the blue. The only difference was that it did not have immobilized enzymes added. So they basically defatted the milk and re-added the same amount of fat as the other samples, but just the fat, not the immobilized enzymes.
I wonder if what, see, like, who's in here, anybody, no, no.
Mhm.
Okay.
Yeah.
Yeah, so even if they defatted, they still have like free lipids because it's a protein. So the defatted milk will have.
Yeah, no. Yeah.
What is, okay, now this is the devil's advocate here. What is the one that they added the nano,
Tubes.
more lipase,
More free lipase?
more lipase, too, the free and the immobilized.
I didn't understand.
So if the defatted milk
Yeah.
doesn't have fat, but
Yeah.
it has endogenous like free lipase.
Yeah.
And then they took that same defatted fat,
Mhm.
the milk, and they put the nanotubes like it. So now you'll have free lipase and immobilized lipase.
Yeah.
coming in. So now we have double the amount of enzyme or iron.
So, I mean, I guess the one thing I can think for that is that the difference between these two is more than double. If it was like,
I don't know. Look at the two fatty acid milligrams per gram.
I'm pretty sure in the paper they quote that it's more than double. So maybe the figure doesn't show it that well. But if it's,
Yeah, but, what's the concentration of the immobilized lipase
yeah,
versus the original concentration of lipase in the defatted? What if they just have more than double the enzyme amount?
so I guess, yeah, no, I know, but like, I'm trying to,
Yeah, like,
no, I know, I just like this paper, so I'm trying to defend it, but I think what I'm thinking is it important for us to know what the free lipase content is if like the increase is more than double, so you know the increase is not just because of extra free lipases, if that makes sense.
Did they add double the same amount of lipase?
So they made a 2%.
Go back to the original,
They
like,
made sure they added an exactly 2% like lipase solution to each sheet milk. So they controlled that. So they're
go back. Also, they did add lipase to the low fat, but they added lipase to the low fat. Spreadsheet.
yeah. So okay, so let me explain. So they have low fat cheese. They have the low fat cheese, and then they made two samples out of it. The first sample they added 2% Pickering emulsion lipase solution, and the other one they just added 2% regular lipase, or like, yeah, 2% regular fat solution with lipase.
So they control how much lipase they added.
Yeah.
Is,
So they did not control how much is still present after defatting it, but they control how much is being added, which is the same amount. Yeah, so that's what I mean. Is like, I think I didn't, so honestly, I didn't think of this in that detail, but I think it still makes sense because that's why I mean, like, if this difference is more than double according to them, so this difference technically is seen by what they're adding and not inherent lipase is still present.
because the not be different in the original defatted milk,
Mhm.
and they took that same original
Yeah.
defatted milk. So they added the same amount of lipase, one with nanotube and one without.
Yeah.
Okay.
Oh, I guess, oh, sorry, was that not clear? Because the methodology section, I thought I covered that. Yeah, so this one. Oh, so I have it in my speaker notes. I don't have it here, but they added an exactly 2 or 3%. Oh yeah, 2%, the same as they use in this small
With
section.
the, lipase free pollution. Yeah.
And like, and on 2%, so 2% free for like the control, quote unquote, and
Yeah.
2% mobilized.
Yeah.
Yeah, so they control them on like, no, they did, okay, so you are right depending, but they did.
Because it made sense in my head. So I thought they didn't miss
Yeah,
it.
because defatting the milk and they are starting with the same defatted milk.
Yeah.
And what, and lipase is a protein, so they won't be impacted by defatting, but anyway, they defatted the milk and it was the starting milk and
Yeah.
just added to that.
Yeah, they just defatted to control how much fat they're starting with.
Yeah, no, it's a good study, except they use toxic compound. That's the only little detail.
I think I figured another devil's advocate's advocate. They did, but did they add the lipase solution to the full fat and the non-fat milk?
No, they did not. I think so. This is why the methodology was confusing, because I wasn't sure what they did with the full fat and the non-fat milk. It doesn't make sense to defat those as well, because then why would those have so much more higher like free fatty acid released? You know?
Because
It's just that.
No, but if they're adding the same amount of fat, like,
the full fat is whatever origin 3.5% fat.
but if they defat it, I mean, like it doesn't make sense to defat the full fat milk and add the same
They
amount.
did.
I don't think they did. Yeah. That part wasn't clear in the methodology, but I'm assuming they didn't. I am assuming they defatted only the low fat ones because that's what they're testing, like the low fat control with free lipase and the low fat with immobilized lipase.
But then that one would have had just native lipases.
The control, yeah. Or no, the full fat, yeah. But because it has so much more triglycerides that it can just keep breaking down more.
Okay, I think we.
Okay.
Good. Okay, thank you.
Thank you.
It is a good paper, very rare to find a good paper. Okay. Well, let's stay with the spirit of milk. And then we're gonna talk about milk proteins. So we have covered lots of ground so far in this map. So we talked about all of the intrinsic and extrinsic factors that will affect protein structure and functionality. Now we're going to talk about the different protein sources and how their structure and how that impact their function. So we're going to talk about milk proteins, the basics, and then as ingredients. And we're going to do the same for all other proteins. So we're going to talk about the native characteristics and how we can produce ingredients from them and how they function. So that's kind of a theme. So we will cover dairy protein, of course, and then we're going to talk about legume proteins, oilseed legumes. So we'll talk a lot about soy and then pea as well, because it is now gaining traction. And then we're going to talk about cereal proteins, given that the gluten is a magic protein, so it's going to have its own part section. And I don't think I'm gonna get to egg. Yeah, but I can put the slides up for you if you want to learn about egg proteins a little bit. But yeah, so and meat proteins back in the day when there were when there was a lot of time and I didn't have a lot of examples, I used to invite somebody from the animal science to talk about proteins. So back in the day. So in terms of ingredients, we have different types in under each category. So in casein, we have the micellar casein, the sodium and calcium caseins. Whey protein, there are so many out there, concentrates, different concentrations of protein. Isolates, soy, we have the flour, the concentrate, the isolate, same with pig. And different kind of categories for the animal-based proteins. And wheat protein, usually you'll find gluten protein ingredient around 70 to 75% protein, maybe tops 80%. So, and we find a lot of hydrolysate ingredients, soy protein hydrolysate, whey protein hydrolysate, caseinate hydrolysate, and also gluten hydrolyzed, egg protein hydrolysate in the market. Also a lot of meat protein, gelatin, especially bioactive peptides, mostly from gelatin hydrolyzed protein. Okay, so we'll start at the very beginning with milk proteins. So the different proteins in milk, so only the amount of protein in milk obviously is very, very low. So you only have around 3 to 4%, depending on the source. What's the moisture content of milk? 87. Yes. So, so the remaining 13% solids, you have a little bit of protein. How much is the percent fat? Simple fat. What is it? 3.5 as well around that? Huh? Okay, depending on the source. We also have high calcium, so we have ash content about what 4 to 5% as well. And then we have lactose. What is it? 3.5? Huh? 4.8. Wow, very good for college board knowledge. Okay. Huh? Yeah, that's good. Okay, so out of this 3 to 4%, seems very, very small, but protein is a big component in not in amount, but in impact. So that milk with 3 to 4%, you can make cheese, which gives you this very unique texture with only having about maybe 3% of that milk. If you have 4% protein, about 3 to 3.2% is casein. So 80% of the protein is casein, 20% of the protein is whey. And then we have a long list of enzymes in milk. The list is long. Very important enzymes that impact structure and functionality of the protein is the proteinases, mainly the plasmin and the plasmin inhibitors and activators. So we call them plasmin system. There's your lipase that we just talked about. Alkaline phosphatase is important just because we use it. You measured it for test and pasteurization. So that's why it's circular. But there's so many enzymes native to milk. That's it for enzymes. We already talked a lot about enzymes. But then if we look at the proteins, so the casein proteins, this is a urea gel. Usually if you run casein on a regular SDS page, they don't separate very well because they're very close in molecular weight. But when you run them in urea gel, you separate them based on size and charge. So you get a little bit of separation. So the alpha caseins, you cannot really differentiate alpha S1 and alpha S2. Very hard to differentiate them on the gel. Beta casein, and then kappa casein. And then this is sodium casein isolate. so it has a lot of the beta and alpha, and you cannot really differentiate kappa. And then this is a hydrolysate casing. So we have four types of casings: alpha S1 and alpha S2 casing, beta casing, and kappa casing. In way, we have beta lactoglobulin is the main one, and then alpha lactalbumin. And then we have minor components: serum albumin, lactotransferrin, and immunoglobulin, but many more as well. But these are the most prominent. Okay, so this is one of the figures that a student made for me out of a very old figure. So, you know, you really want some extra points, and you see a shabby table, and can help out. Okay, so this is a distribution of milk proteins. We have the minor proteins, we have the enzymes, and then we have the casein. You'll see here alpha S caseins. We have S1 and S2, and there are different, the A, B, C, D, different genetic variants of them. And then we have the kappa casein, and then beta casein is unique because you have intact beta casein, but you also have fragments of beta casein because of the plasmin and endogenous in milk. Over time, the beta casein partially can be broken into three different segments, depending, because plasmin has the same kind of specificity to trypsin, so it cuts after lysine. So there are unique spots where the plasmin cut, and it gives you three gamma caseins. And proteose peptone, I think we talked about it in college on Tuesday. Protease peptone is what remains from when you cut and you get the gammas. You have a very soluble peptide because it's a beta casein is very amphiphilic, meaning the regions of hydrophilic and hydrophobic are very well separated. So when the enzyme plasmin cuts, there is a hydrophilic peptide that ends up in whey. So because it's soluble. So when you separate casein and whey, you're forming curd, and then in the whey, you will get some proteose peptone because of it being soluble after you cut. After plasmin hydrolyzes the beta casein. So that's what these are. Plasmin is heat stable. And even after pasteurization, you still have plasmin activity, and it still contributes to breaking down of the beta casein. And also it's important during ripening because not only fatty acids contribute to flavor, what else contributes to flavor? Yeah, protein, amino acids, and small peptides, basically, they contribute to taste and in general flavor. So yeah, plasmin plays an important role in flavor development. Okay, so and then in whey we have your beta lactoglobulin as a major whey protein, followed by alpha lactalbumin and the other minor ones, the immunoglobulin, serum albumin. Bovine serum albumin, we use it in the lab, so that's where it comes from. We use it as standard most of the time. So this is the serum albumin. Let's see, again, there are different genetic variants. So you will see those letters as an indication of genetic variants, and those genetic variants vary a little bit in the amino acid composition. And there it's like differences, substitution of some amino acid or one or two extra amino acid. So just a slight variation. Okay, so we're going to talk about the characteristics of these casings, and how they differ. So all of them, alpha, S1, S2, and beta casein are phosphorylated. So I think it's the threonine that is phosphorylated. I might mix them up. Threonine, phosphorylated, and in kappa casein it is, no, the other way around. Serine is phosphorylated and threonine is glycosylated. So kappa casein is glycosylated, and the alpha S1 and S2 and beta are phosphorylated. So they are the ones that bind calcium. So the calcium phosphate bridges are due to these casein. Beta casein is the most hydrophobic and the least phosphorylated. Alpha S2 is the most phosphorylated, the most hydrophilic. That's how they differ. And because they are phosphorylated, they are very sensitive to calcium. So you can precipitate them by adding calcium. So kappa casein is glycosylated, and in general, the caseins are open structured because of toluene. The secondary structure breaker, 70% on average is the percentage of toluene compared to other amino acids in casein, which makes them hard to bend and structure them into a globular protein. So they're highly amphipathic or amphiphilic. Amphipathic and amphiphilic are used exchangeably. They mean the same thing. They have distinct regions that are hydrophilic and distinct regions that are hydrophobic. So which makes them natural emulsifier. Because now you have an open structure, you have a lot of surface activity, surface hydrophobicity, surface hydrophilicity, and ability to orientate at the interface readily or absorb at the interface readily. Interface of oil, water system. Okay, so what I want you to know is not really, don't memorize the number of phosphate groups, for example. You want to know that they are phosphorylated. Alpha S1 is phosphorylated, but alpha S2 is more phosphorus. So maybe that's what you want to remember, is alpha S2 is more sensitive to calcium than alpha S1. So, and that phosphorylation adds to their net negative charge. And they have, like I said, clusters. So the white is the hydrophobic. The striped is negatively charged, and the gray part is just polar, but not so. That's how I simplified the drawings of these cases. So you see they have clusters of hydrophobic, clusters of charged, polar groups. Alpha S2, it has the highest amount of phosphate. That's what I want you to remember. I don't want you to remember 10 to 13 because it's the most phosphorylated, is the most sensitive, obviously, to calcium precipitation. Because it's the most phosphorylated, it's the most hydrophilic. I obviously don't want you to remember numbers. It's just, it's unique in a sense that it has 3 clusters that are negatively charged, many clusters that are polar, and a huge portion on the C terminus that is hydrophobic. And one small pouch here that is hydrophobic. So it is very characteristically different than alpha S1 in terms of the distribution of its different clusters. And what is also unique, I don't want you to remember necessarily the net charge. So don't worry about those numbers, really. What I want you to know that it has two cysteine residues. So alpha S2 and kappa casein are the only caseins that have cysteine in them. And but only two residues each that form disulfide. Beta casein is the most hydrophobic and the least phosphorylated, and it's very, very amphipathic or amphiphilic. Look at the long carboxyl end that is hydrophobic and just a little tail that is hydrophilic phosphorylated. Very sensitive to temperature. If you remember, we said that hydrophobic interactions are weakened under cold temperature. So that's why in refrigerated milk, the whey component has some beta casein that reach out of the casein micelle because hydrophobic interactions are weakened. And beta casein is in the micelle interacting mostly via hydrophobic interaction because it's not as phosphorylated, so it doesn't form as many calcium phosphate. So it's interacting in the micelle with the other caseins via hydrophobic interactions. And that's why it moves it out of the casein micelle during the refrigeration of milk into the whey part. That is very unique for beta casein. So it has the highest proline content, very random structure. And this is what I said about it being sensitive to plasmin hydrolysis. And the hydrolysis releases 3 distinct gamma peptides and the very hydrophilic, this proteose peptone that goes into the whey component because it's very soluble. That's why sometimes whey, that's a good college board question, has proteose peptone and one other thing from casein. Whey has. So what happens during diet? Yeah, glycomacropeptide that is part of the kappa casein that gets hydrolyzed by the chymosin or rennet enzyme. So in the whey fraction, sometimes we will have the proteose peptone from beta casein and the glycomacropeptide from the kappa casein, especially in cheese whey. So that's why whey protein concentrates that are isolates that are produced by membrane protein will have these components. But whey protein isolate that is produced by ions from agripore will not have these components. So only the whey protein produced by membrane filtration will have some casein peptides. Kappa casein is also very amphiphilic or amphipathic because it also has a cluster from the N terminal, so they're kind of like opposite. The beta casein is C-terminal hydrophobic, but the kappa casein is C-terminal hydrophilic. And that component is glycosylated by the threonine residues. So that's why it is negatively charged due to glycosylation, which happens during post-translation modification in the cells. So, and it is also unique that it has the 2 cysteine groups that I talked about as well. So this is the one that gets torn or hydrolyzed by the chymosin enzyme. So this is the glycomacropeptide, and this is the known as the para-kappa casein. Para-kappa casein is the hydrophobic component that stays with the curd, with the cheese. Okay, this is not sensitive to calcium. There might be one phosphorylated group, one phosphosyl group that is of insignificance in this study. I think that's the most important thing. I don't necessarily want to know which carbohydrate is attached, but galactose is one. This is just a summary drawing, just to kind of look at the differentiation between those four cases. Beta and kappa are easy to remember. They are very amphiphilic, with one portion that is negatively charged and another portion that is very hydrophobic. Alpha S1 and alpha S2, they have their charge groups in the middle of the protein sequence. Also very amphiphilic because they are clustered. The charged and the hydrophobic group are clustered, not distributed evenly. Yeah. And you can see they're very similar in the number of amino acids. That's why they're really very close in molecular weight and hard to see on SES paper. It'll have to leave you a little bit. So because they are phosphorylated and glycosylated and very hydrophobic regions, they form micelles. So just general characteristics, but really I don't want you to know much about the numbers here other than the ratio of alpha S1, alpha S2, beta, and kappa. The concentration of kappa casein in particular is directly related to the size of the micelle. The more kappa casein you have, the smaller is the micelle, because I'll tell you why. It's mostly on the surface due to that glycomacropeptide region. So the more kappa casein you have, the smaller are your micelles. But in general, the ratio is very similar, varies with sources and genetics. The casein micelle. So there are different models that have been developed for understanding the casein micelles. Oops. The original model that was proposed early on in the 70s is the core model. The next model that came after that was the subunit, submicelle. And the last model is the nanocluster model. Which one do you think is most accurate? What do we know? What do we know? Anybody? Huh?
Everyone says that they are wrong.
Yeah, but everybody says that their model is correct, but the most accepted model is the nanocluster model. We'll talk about each one. The core model, just because of what we know about the kappa casein, it was assumed that the kappa casein are only, all the kappa caseins exist on the surface of the micelle. And what all the others are entangled in the middle of the micelle. That was the original theory. And that there is a linear relationship between micelle surface area and kappa casein. So what is correct about this model is only the last point. Now, after that came the submicelle model, where the casein micelle was thought to be made up of submicelles. Each of these circles is a submicelle, where the core is hydrophobic and there is a little bit of the kappa casein on the surface. The submicelles that have the highest concentration of kappa caseins are on the surface. That doesn't mean that the submicelles in the middle does not have at all any kappa casein. They do, but very low concentration. The the ones on the outside constitute most of the kappa casein. And then the way they are linked, so these are like hydrophobic core, and then the phosphate are causing calcium phosphate bridges. So we have calcium phosphate cluster, and then the submicelles are linked together by those calcium phosphate bridges. So this was kind of confirmed when looking at a microscopic scan, and they saw there's this little, polka dots, and they say, "Oh yeah, okay, so this is really submicelles." That is not correct, but that's how they confirmed the submicelle theory by just having these little dots shown. So here's just an illustration, bigger illustration, that this is your calcium phosphate cluster, and this is your serine residue on the alpha S1, S2. And then they are linked to another casein via these calcium phosphate bridges. Now the nanocluster model, there is no, yes.
So, what was wrong about this model then?
Okay, so the imaging was misleading. So when they saw the imaging, they saw these dots and they say, "Oh, okay, so we have submicelles." And then they started theorizing how these submicelles are formed. It was based on theory and reading wrong the image.
So, but the fact that more is in myself are on the outside, that is true.
That is true. Yeah, because of the fact that this is the glycomacropeptide that is very negatively charged, is stabilizing the micelle, so that making that colloidal system, otherwise the protein will have test. Okay. So then the nanocluster model said, no, you know what? Okay, kappa casein is mostly on the surface, but there are some kappa casein in the middle. And all of the rest, the alpha S1, S2, and beta are really entangled in the middle of the micelle and associated together via hydrophobic interactions and calcium phosphate bridge. So all of them have this common correct thing, is the kappa casein is stabilizing the micelle. So basically, so many people drew the micelles and theorized the micelles, but in different ways. So this is just an illustration from a reference that I have on Canvas for you. But this last one here is the one that is most accepted among researchers. And when they did microscopy again, it looks, as if it is dotted, but that dark spots is actually your calcium phosphate cluster. So it's not submicelles. That disruption is actually the calcium phosphate cluster. Yep. So if this is an animation of what the actual micelle would look like. So what you have here in gray is your calcium phosphate clusters. The green here is your para-kappa casein, the hydrophobic component. And then those little hair-like structure is your glycomacropeptide. Those orange are your alpha S1 and beta casein that are linked via the phosphate cluster. And the blue is the very hydrophobic component of beta casein that is linked to the other caseins via hydrophobic interaction. So this is kind of the animation of the nanocluster model and how the proteins are interacting. Huh? The blue is just to illustrate that beta casein being very, very amphiphilic, that blue part is the hydrophobic component of beta casein, which does not interact with the phosphate, calcium phosphate cluster. It interacts with the other casein, the hydrophobic. So basically, the casein in my cell is stabilized by three main things. One, calcium phosphate bridges, hydrophobic interactions, and the glycomacropeptide on the surface giving it the negative charge that causes the micelles to repel each other and stay in a colloidal system in your neck. Okay? All right. Yes. Yeah, the three wait. So the casein in my cell is stabilized by three important forces. One is the calcium phosphate bridges. It's mostly in the inside of my cell. The hydrophobic interactions involving beta casein, and then the kappa casein on the surface with the glycomacropeptide, giving that negative charge that allows the micelles to repel each other and stay in colloidal system. Okay, so three forces. So casein is very thermally stable, but it's not like thermally stable at all costs. It does get destabilized with extensive heating for an extended period of time. So what happens during that, a lot of things happen during heating. First thing that takes place as we're heating, whey protein building. The first. Why is that important? It's important because that sulfide group that is free in whey protein, beta-lactoglobulin, can then induce disulfide linkage with catechols. That's the first thing that can happen. Beta-lactoglobulin denatured exposes a sulfide group and cause disulfide interchange and linkages with catechols. The pH drops, and I'll show you why there's different reasons for dropping of the pH. One of them is the degradation of lactose into lactic acid. A precipitation of calcium phosphate. We form colloidal calcium phosphate, which releases protons. And when you form bigger cluster of calcium phosphate, you release more protons that causes the pH. Casein with heat gets dephosphorylated. We lose even more net negative charge. Kappa casein get hydrolyzed because we get this dephosphorylation, we decrease charge. And then hydrophobicity is increased when the charge is decreased, and then we get aggregate via hydrophobic interactions. And then the colloidal calcium phosphate will actually start forming bridges between different micelles. So enhancing the aggregation aspect. And this is illustrated here. So I illustrated what happened, maybe in a visual way that is a little better. So colloidal calcium phosphate increase. So that means we are destabilizing the calcium phosphate bridges within the micelle. And when the colloidal calcium phosphate increase, so we have large entities of calcium phosphates, colloidal, release protons. So these protons that are released obviously become more acidic, so we decrease the pH. Breaking down of lactose also decreases the pH. Breaking down of phosphoserine, so dephosphorylation, releasing protons, also decrease the pH. when we decrease the pH, we are getting close to the isoelectric point. So we're lowering the zeta potential to the negative charge on the surface of the casing. And these colloidal calcium phosphate now form new interactions between micelles or among the micelles rather than within the micelle. All of this will cause aggregation. So when we lose charge, we have hydrophobic interactions, we have new colloidal calcium phosphate bridges, we have beta-lactoglobulin that is denatured, forming disulfide interchange with kappa casein, all leading to aggregate. So this all happens when you extensively heat. Okay, so actually lactose converts to a lactic acid.
Oh, the elimination of lactose positive.
Yeah, yeah. Yeah, so the arrow here, so the lactose decreases, degrades, causing more reduction in pH. So this is kind of like visual to that chapter that I put from Fenema on daily proteins. Yeah, it's so complex given how smart Fenema is, but I just broke it down into the graph. But if you want to know more, and go back, it's all written in that chapter that I underlined where to read. So, yeah, my old fashioned. Print the book chapter and underline. Okay. Yeah, a hydrophobic interaction, obviously, because of low charge. All right, so this is how irreversible aggregation happens. Also, casein micelle is they are susceptible to acid, and that's how yogurt is formed. When we reduce the pH due to fermentation and production of lactic acid, during the fermentation, the pH reaches 4.6. Zeta potential is lowered to 0, and casein aggregate with no fusion, meaning you form a gel. You entrap the water, and yogurt is a kind of gel. Wheat, yes, but it entraps water. And syneresis happens over time. This. Whereas casein form a coagulant because of enzymatic activity. It is sensitive to chymosin degradation, which is really cutting off the glycomacropeptide. The casein and micelle fuse together via hydrophobic and again calcium bridges. That's why calcium is added to milk that is going to be used for bird formation to enhance the yield. And that's another form of destabilizing the micelle. This is a nice illustration of a stable micelle in the milk, in the fresh milk. You have stability and repulsion between the micelles because of this glycomacropeptide. Here you have hydrolyzed off, broken off the glycomacropeptide due to enzyme activity. Then you start to form the curd via calcium phosphate bridges and hydrophobic interactions. Here in C is pH at the isoelectric point. So this glycomacropeptide loses its charge. So now we form the yogurt. And in D, actually, this is a denatured, heated milk, where this blue is your beta-lactoglobulin that form disulfide linkages with kappa casein, which enhances the gelation. Sometimes they add whey protein to yogurt just to enhance the gelation power and reduce humidity over. And that's because the beta-lactoglobulin interact and enhance more gel formation and water retention. However, if you heat the milk to an extent that you cause this beta-lactoglobulin linkage to kappa casein, and we try to form to make cheese with chymosin, it won't work it, because now the chymosin is hindered from reaching the breaking point of glycogen. That's why milk cheese is pasteurized at lower temperature, like 16, 20. Yeah. So that they don't denature beta-lactoglobulin and don't cause that linkage, because otherwise the enzyme won't work as well. So over time, milk, pasteurized milk or even sterilized milk will lose quality because you will see protein sedimentation and separation. We call that age gelling. So over time, pasteurized milk, you'll have your beta-lactoglobulin denature. So over time, it will start interacting with casein, and we lose surface charge over time, and plasmids break down beta casein. Then we start seeing thinning, and then precipitation and gelling. So we call it thinning and gelling. The milk separates. Okay. Any questions on these? Well, it's not like it's just get the light on, the light off. It's kind of like a process, but ultimately above 140. Everything start, if you eat protein for eating at 40 degrees, you are starting to break down and you're making changes, but completely elevation will not happen until you reach a certain point. But of course, at lower temperature, but extended exposure, cannot continue. So temperature time combination basically is important. All right, so whey protein. So whey protein is completely different than casein protein in how they associate. Mostly, okay, without looking at them, does whey protein beta-lactoglobulin, does it exist as a monomer or dimer or octamer as neutral? And not everybody know this. People think it's monomer, but it's actually a dimer at around neutral pH. If you don't know that, and you're running size exclusion chromatography and you see beta-lactoglobulin at 32 kilodalton, you know what happened with my 18 kilodalton beta-lactoglobulin? Well, you won't see it because if you run a phosphate buffer, it's a dimer at pH 7. Okay. So this is alpha-lactalbumin. It has mostly alpha helix with one beta sheet fraction, whereas beta-lactoglobulin has a lot of beta sheet and one alpha helix. They are globular protein compared to casein that's open structured. And then we have 5 cysteine residues, and 4 of them are form a disulfide. One here, and then you have this free sulfide group, which is very essential, very reactive. Once the protein denature, if you have alpha-lactalbumin, that all of the cysteine are SS, this free sulfide group will act as a reducing agent and promote disulfide interchange among all of the beta-lactoglobulin and alpha-lactalbumin combined. So it really is very important group. It's that free sulfide group. So highly structured, globular, minimum flexibility because of the disulfide linkages that keeps that protein really tightly globular. Alpha-lactalbumin denatures at 63 degrees, beta at around 73. These are average temperatures. And the reason that alpha denatures at lower is because of alpha helix is more thermally labile than beta sheet. So beta-lactoglobulin, like I said, is mostly beta sheet. The secondary structure, 5 cysteine residues, 1 short alpha helix. It is very, very globular in the sense that most of its hydrophobic residues are in the interior. And it's very hydrophilic on the surface with minimum hydrophobic groups on the surface. So surface hydrophobicity of white protein is really low. So it is like 1500 if you're familiar with surface hydrophobicity, let's say it is 1500 and P is 15,000. So it's like with P protein, but 10 times more hydrophobic on the surface than white. So that is it. Okay, with that, because of that, if you remember when we talked about solubility, functionality of whey protein is so unique that if it's not denatured, it's soluble at a wide range of pH, even at its isoelectric point, which is about 4.5, 4.6. So even when it doesn't have net charge on the surface, it doesn't have enough hydrophobicity to bring the proteins together. And the SH group is hiding inside. So it's just not gonna aggregate. Now, when you heat it, however, then it's gonna precipitate around 4.5. Why? Because once you heat it, it's denatured, you expose this SH group, you expose the hydrophobic group, then you get protein. Then it's not soluble at the isoelectric point. It is I think maybe the only protein that does not precipitate its isoelectric point because of how hydrophilic it is on the surface. I don't know any other protein that does that. So of course, it is sensitive to heat. The natures on average at 72 degrees, and that causes disulfide interchange. And also it causes, if it's in the milk system, it causes it to interact with casein. And if we want to make cheese out of that milk, we're going to have problems because it will interfere with chymosin. So it is pH dependent. So it's most sensitive at pH 4 or 4.5. If you heat the protein at that pH, it will quickly sediment and coagulate. It has good stability at pH 6, and then stability decreases at alkaline. So if we want to look at what they form at different pHs, so dimer around neutral, but then there are monomers as you increase or decrease the pH. If you increase the pH above, way above 8.5, then they would denature and fall. Around the isoelectric point, they form octamers. Because, you know, you're starting to lose the charge. Question? Alpha-lactalbumin, we talked about it. It is mostly alpha helix structures, 1 beta sheet, 8 cysteine residues, also very compact. It binds calcium, and calcium helps, the binding of calcium helps it to be stable. And it has a function, which is lactose synthesis enzyme regulator. That's its biological function. So more heat sensitive than beta-lactoglobulin because of the alpha helix structures. The denaturation can be reversible if it's not exposed for a long time. And if we lose calcium that is bound to it at low pH because it leaches out, then it becomes more heat sensitive. So these are kind of characteristics. Now, this is an old table from an old pathetic book. Yeah, if you are using alpha-lactalbumin alone without beta-lactoglobulin, you won't see disulfide. You'll get demonstration and the photo interaction, but you don't have anything that stimulates disulfide. That disulfide group acts as a breaks, and so it reduces and oxidizes, causes the reduction and oxidation, causes the injection. Oh, it depends if you heat, you are gonna cause the inflammation and agitation. So there is a combined effect of the pH and temperature. But if the protein is in regular solution, no heat, but you can the pH, it will be either monomer, dimer, or oxygen. When you include the heating, then you have that interaction. pH temperature interaction. So that's this table. It kind of tells you the difference, it kind of a comparison between whey and casein, and within each, how beta-lactoglobulin is different than alpha, different than bovine serum albumin, and how the different caseins are different in molecular weight, in total number of amino acids, in percentage of hydrophobic residues, and number of protein, and so on. But here's an exercise we're gonna do together. All right. Which you might see in your next evening. Okay. So if I wanted to differentiate casein from them, and I say, okay, how are they different in amino acid distribution? What do I need? No, not in content. In location, distribution. If I tell you, then I answer. There's no way of telling. Huh? What? But not the composition, I'm not asking about it. Think of sequence, distribution within the sequence. What? Okay, I give up. Yes, yes. So highly amphipathic and regular. Yeah, so casein has very distinct clusters. That's what makes them surface active. Whereas whey protein, they are randomly distributed. You don't have how to, very amphipathic. Okay, molecular structure. Open structure, globule. All right, open, random coil, little secondary and tertiary structure. Okay. What about heat stability and gelation? Casein is more heat stable. Yeah, it gels in the over aging, enzyme gelation, acid gelation. Okay. Whereas whey is heat labile, heat induced gelation. Associate. What do casein form? So how do they associate into? They form? So whereas whey does not, it's dispersed, monomer, dimer, octamer, basin. Enzyme sensitivity? Huh? Okay, so is enzyme sensitive to chymosin, to plasmin, other proteases? The whey protein, non-binature, resists hydrolysis. That's why babies, they put in infant formula hydrolyzed whey, because their digestive system is not yet developed. So they cannot digest whey protein very well. So in the infant formula, they put hydrolyzed whey protein, and it is very bitter. Doesn't taste very well, but poor babies, they can't talk. I tell you, can you not give me this formula, please? Okay. Surface absorption and emulsification. So which is more surface active? Casein. Flexible open structure, high surface absorption, low surface absorption in a native state. Solubility, why did I click it? Okay, so precipitate at pH 4.6. Whey protein is soluble over a wide range of pH when not heated, even at the isolate. So, conjugation. Is casein conjugated? Well, phosphorylation, glycosylation. But whey does not have any conjugation. Sulfur residues, high, low in casein. You have one SS in kappa, one SS in alpha S2, whereas whey protein is rich in sulfur group. Okay, so that's it. Yes, taking a picture. Let's say if you're taking exam like that. Okay, I don't know, I'll have to check. Okay. So functional properties, as we learned, they are related to molecular characteristics. So what are the molecular characteristics? Just a review. We studied that for the exam. What are the molecular characteristics that impact functionality? Let's review. Okay, what else? Okay, configuration. Amino acid composition. What else is important? Size, surface properties, a bunch of them, right? It's just like charge, surface activity, configuration, size. You can add amino acid composition, all of that. So we're going to take that into account as we talk about the functionality of these caseins. So casein, natural emulsifiers, so they have surface, they're very surface active, they readily absorb at the interface. What does that mean? You have oil, water, and you're homogenizing. Caseins readily orient themselves in a way that the hydrophilic component towards the water, hydrophobic towards the oil. So that's what it means, readily absorb at the interface of the two phases. Unfold, well, they are or kind of already unfolded and rearranged themselves and start to form that film around the droplet. So beta-lactoglobulin, on the other hand, does not have good surface activity because of very low hydrophobicity on the surface, so you don't have good balance. So native state, forget it, they're not gonna emulsify. If you denature a little bit, open up the structure, then they can start to improve the hydrophobic to hydrophilic proportion on the surface. At low pH, it's compact, it's rigid, it's not flexible. Around the isoelectric point, you improve things a little bit, you have less repulsion, you can improve a little bit the absorption. And if you reduce the disulfide into this, then you can also enhance, when you are homogenizing, you can enhance unfolding better and better. What I should have had here is when you denature by heat, which I didn't, or physically. So, yeah. All right, so gelation of casing, we talked about enzyme gelation, pH gelation, heat coagulation. We talked about all of that, and H gelation, because over time you lose a charge, and you have native enzyme activity that causes gelation over time. And beta-lactoglobulin denatured in pasteurized milk. Now, beta-lactoglobulin is a very good gelling agent in nuclear. So actually you can form, by using our method in the lab with gelation, you can form a permanent gel with whey protein at 5%. So, whereas if you are trying to form a gel with whey protein with love, you might need 20%. So you would think it's a small, very soluble protein. How come it forms good gel? It forms good gel because the nature is very good during heat, when you induce it, and you have that mischievous stuff, sulfide group there. When you denature the protein and expose hydrophobicity, and you have that reacting group, you very well can cause good gelling behavior. So, but also it is dependent on pH. So if you have pH around the isoelectric point, it's not that great of a gel, it's opaque, it's rigid. But if it's away from the isoelectric point, you get nice transparent elastic gels. But if it's very high pH, you don't form a gel because of the repulsion, basically. If it's around the isoelectric point or close, you really need to heat above the denaturation temperature, or before, sorry, the denaturation temperature, because you don't have as much charge, so it is sensitive to temperature, and you can form gel under the denaturation. And if you carry charge, more charge, then gelation starts after the denaturation. So there's always the message here is pH temperature interaction. So it is very dependent. The thermal denaturation and production of a gel is very dependent on the pH as well. Heating rate is important. So 10 degrees per minute is high heating rate. 1 degree per minute is moderate heating rate. So if you are at a close to isoelectric pH, you don't want to heat very fast. You're gonna get aggregation because you already don't have a lot of charges and you're heating very fast. The protein doesn't have time to form nice network. It will aggregate. So high rate of, in general, high rate of heating cause precipitation, especially around the isoelectric. So ionic strength, the salting in, salting out. So a little bit of salt may enhance gelation, but a lot of salt cause aggregation and precipitation. So disulfide interchange, if we block SH group, we reduce gelling ability. If we add cysteine, a reducing agent, you actually It enhances reduction and oxidation, so it enhances disulfide. So protein concentration is very important, whether it's whey protein or soy or pea. The higher the protein concentration, the stronger is your gel. But in terms of PLZ, we can form a gel around the isoelectric point. Even 1% can form a gel. But away from the isoelectric point, 5% gel can be formed. So now we'll look at production of milk proteins. Yeah, I can spend a few minutes and finish that. We're almost done. So you take the milk. I don't know why skim milk. It doesn't have to be. But oh, skim milk. You have really bad tasting cheese, like she was telling me. Anyway, so you have the rennet casein, that's your curd, and then you have your sweet whey. And then if you are producing, reducing the pH and precipitating the casein, then you can produce acid whey, and then you take that curd or casein that you precipitated, you neutralize with either sodium hydroxide or calcium hydroxide. You get sodium caseinate or calcium caseinate. Now you have the whey, mostly it's sweet whey that we usually produce whey protein isolates and concentrates from. If you take the whey and just heat it, you get lactalbumin, which is a heat aggregated whey protein that doesn't have any function. It has good nutrition, that's all. If you take that whey and then concentrate it by membrane or ion exchange, then you get whey protein concentrate and whey protein isolate. If you take that milk and you just remove the fat and then heat it with calcium and reduce the pH, you get a co-precipitated casein and whey protein. And it's just used as enhancing of nutritional part for nutritional purposes only. So this is how we produce whey protein ingredients using membrane. So you have the cheese, you get the whey as a byproduct of cheese production. You run it through ultrafiltration. The retentate is your whey protein, and the permeate is your whey permeate that has a little bit of protein and a lot of lactose and minerals, other solubles. So the retentate can be either 34% protein, 50% protein, 80% protein you'll find in the market. What causes this concentration? What determines if you're getting a 34, a 50, or an 80? Huh? Yeah, so continuous washing, multiple passes. Then depends on how many passes and how many washes you can concentrate the protein. That's why it's more expensive. So the WPC 80 is more expensive than WPC 34. Okay, I want to produce an isolate, then I take that and run it through microfiltration. So you would, it's kind of counterintuitive, but this is how it happens. Ultrafiltration gives you whey protein concentrate, and taking that retentate from here and running it through microfiltration, you are now capturing phospholipids and aggregated protein. So now what goes through that, the permeate, is mostly protein. And that's why it's whey protein isolate, or it's 59%. So just differences here in characteristics. So the lactic acid casein, if you don't add any sodium hydroxide or calcium hydroxide, it's insoluble at 4.6 or 4.7, obviously around its isoelectric point. It's low in calcium and phosphate. They leached out. Solubilized, you can solubilize it by increasing the pH. Rennet casein is insoluble at pH 7, it forms an aggregate. It's high in calcium and phosphate because calcium phosphate breakage is what stabilizes the curd. And if you want to solubilize it again, you can sequester calcium. And you can this way break the calcium phosphate. Heat stable, good flavor, good nutritive value, PDCAS of 1, because in PDCAS we use 18 acid acid. Complete amino acids, highly digestible, and it's used in applications like cereals, breakfast cereals, snacks, baked goods. Now, if you take that acid caseinate and neutralize it with sodium caseinate or calcium caseinate, sodium hydroxide or calcium hydroxide, you get calcium caseinate and sodium caseinate. They're very different in characteristics. Calcium caseinate does not form to solution. So it's not for use where you need water binding or viscosity building. You don't use that. Has very low viscosity, it's opaque, it's used in applications such as creamers, wood topping, imitation. Whereas sodium caseinate form true solutions, and it's good for emulsification, enhancing viscosity, it's translucent and used in many applications. Whey protein, good source of sulfur-containing amino acid. You can get it from acid or sweet whey, mostly sweet whey. Lactalbumin doesn't have any functionality. It's denatured, aggregated, insoluble, used only for nutritional enhancement. Whey protein, different ways of isolating, IE for ion exchange. Highly soluble, used in so many different applications. The golden standard for protein ingredients. Oh, we're done. Just 3 minutes more of your time. All right, we're done with dairy. Next week, legume proteins. Forward. What? Oh, my battery is low. Good timing.